Respected teachers and my dear friend, myself Aniruddha Satish Deshpande, research student from Department of Botany, Government Vidarbha Institute of Science and Humanities Amravati, and I am here to present a paper on the topic Evaluation of Cytotoxicity of Common Vegetables Momordica charentia and Lagenaria cicereria by Allium Test System. So starting with introduction, vast resources of medicinal and aromatic plants which are available worldwide. They are Till now, they are used as a traditional medicines. About 80% of the world's population depend on it. From ancient times, many Indian medical practitioners made use of common vegetables and medicinal plants for curing many devastating diseases. Such two plants are Momordica charentia and Lagenaria cicereria, which offers many medical advantages along with the source of some important nutrients. But when they get consumed raw by rural poor or even the urban people, they provide many side effects. So there must be distinction between health promoting and toxic doses of these two vegetables. Allium test system serves as an efficient tool for that, which is given by Jared Fiskesjo. It involves direct treatment of onion bulbs with plant extract of such substances suspected of being capable of causing cytological defects. It is assumed that defects occurring on treated onion bulbs can also be expressed in human cells. So it is very much effective for cytotoxicity studies. For this, the two different vegetables, that is Lagenaria cicereria and Momordica charentia, were bought from the market of Amravati. Then the extract was squeezed out from them by using 1 liter of clean, ordinary tap water pounded in mortar and pestle with the fruits. For allium cepa assay, healthy bulbs of equal size were germinated over water before being transferred to each of the test plant extracts, and onion with new root tips were placed on coupling jars containing the plant extract for about 3 hours. After 3 hours, the root tips were harvested and they were first fixed in cornice fixative 1 for 24 hours and after that in 70% alcohol till their use in cytology. For squash preparation, root tips were hydrolyzed in one normal HCl for about 7 to 10 minutes. The squashing was done over 2% acetoarsine stain. The slides were then studied under the high magnification microscope and the total number of dividing cells, non-dividing cell, mitotic index and percent chromosomal aberrations were counted and recorded. Results and discussion. So study determined the cytotoxic and genotoxic effect of extracts of two different medicinally important vegetables. Table 1 shows the effect of different concentration of Momordica charentia fruit extract on mitotic index and abnormality percent of Allium cepa root tips. So for this table, the graphical representation, as you can see on the screen, that the mitotic index goes on decreasing with increase in concentration of these extracts. The control shows the highest value of mitotic index, that is 20.13, while the least value was observed in the highest concentration of the extract of Momordica charentia fruit, that is 12.53%. Whereas as you can see, the abnormality percent is 0 in case of control, but it increases linearly among the concentration of this extract with highest abnormality percent, that is 15.15% in the 0.015% extract. Similar trend was followed in case of Lagenaria cicereria. As you can see on the screen for this table, the graphical representation, as you can see on the screen, that is figure 2, which provide the effect of different concentration of Lagenaria cicereria fruit extract on mitotic index and abnormality percent of Allium cepa. As you can see, the control shows the highest mitotic index and the value of mitotic index is linearly decreased among the increasing concentration of the extract of this Lagenaria cicereria fruit with least value of mitotic index, that is 15.73% observed in highest concentration, and the similar trend about abnormality percent was followed. As you can see, in case of abnormality percent, it matches to the Momordica charentia, that is the highest value of abnormality percent is in the highest concentration and control shows the 0% abnormality value. Normal mitotic phases observed were interphase, prophase, late metaphase, late anaphase, telophase, late telophase, whereas mitotic abnormalities were observed in the form of clumped prophases, clumped metaphases, unorganized metaphases, sticky anaphases, bridges, multiple chromosomal bridges, and single chromosomal bridges. Along with that, laggard and precocious movements are there. So earlier, many workers use allium cepa test in order to determine the cytotoxicity of different chemical as well as biological compounds. Summary and conclusions. So, the cytogenetic test serves as an effective tool for identifying damaging effects of various substances. The allium test is useful to evaluate genotoxic, cytotoxic, and potential of medicinal plant. The decreased maturity index suggests that the presence of cytotoxic substances is there among the test substance. Along with that, the chromosomal aberration were due to the blockage of DNA synthesis and inhibition of spindle formation. Different mitotic abnormalities may be due to the different phytochemical compounds like alkaloids, flavonoids, terpenoids, tannins, and carcinogens, etc., which are present in the plant. And since most of the herbal preparations are not refined and they are mostly consumed in a raw form, this suggests that the possible exposure of the cells of the consumer to such genetic defects. Studies suggest that even at the lowest concentration, that is 0.005%, plant juices were found to be cytotoxic, which in turn suggests that daily raw intake of this plant must be lower than 0.05%. 0.005% in order to avoid any cytotoxic effects on human health instead of being beneficial. So study provides an important basis for further investigation of crude extract of the same two different medicinally important vegetable plant for cytotoxicity and genotoxicity analysis. This study suggests that the higher dose of the extract may be harmful for human health, which needs further animal experimentation. References. These are my references. Thank you.